Hello friends, this video on Cell, the Unit of Life, Part 25 is brought to you by examfear.com. No more fear from exam. So when we talk about prokaryotic cell, we don't really need to talk much about it because they again do not have any membrane bound cell organelles. However, they do have the genetic material. So now whatever part they have, we will quickly discuss about them. So first we will look at the characteristics of a prokaryotic cell. So as the name says, pro means before, karyon means nucleus. So this was the cell which existed before nucleus. So when nucleus, the cell without any nucleus. So membrane bound organelles are absent here. You do not see any mitochondria, any Golgi body or any lysosomes here. Cell compartmentalization is completely absent. So the cell is not divided into compartments. So it is something like everything is scattered inside the cytoplasm. The genetic material or the proteins, the enzymes, everything is just scattered here and there. Membrane bound nucleus is absent. Now, in case of eukaryotic cells, nucleus was bound or surrounded by a nuclear membrane, and inside that, the genetic material was properly organized. But in this case, there is no membrane bound nucleus. However, genetic material is present, but that is just scattered in the cytoplasm. Genetic material is present, as I mentioned just now. Now, where do we see this prokaryotic cell? It is seen in the kingdom Monera, where we talk about bacteria, and it is also seen in archaebacteria and the blue-green algae. So those are the organisms where a prokaryotic cell exists. So in this case, all intracellular components, all the cellular components are located together in the same volume enclosed by the cell membrane. So when you talk about the shapes of prokaryotic cells, again they exist in a variety of shapes which we'll see very soon. For example, if you talk about bacteria, they can be rod shaped. When you talk about the mycoplasmas or you talk about the blue-green algae, so not all of them have the same shape. So these kind of cells can exist in a variety of shapes and sizes. So now we will discuss about the structure of a prokaryotic cell, even though the structure is now not going to be that complex. So it has a cell envelope. So here the cell envelope which we have is little different from what we had in case of eukaryotes. In eukaryotes, what all did we have as part of the cell envelope or cell covering? We had a cell membrane which was common to both plant and animal cells, and we had a cell wall which was present only in plant cells. Here, as part of cell envelope, we have three layers, that is glycocalyx, cell wall and cell membrane. Now we will discuss about each of them in detail in the next slide, but these are the three layers which are present as part of the cell envelope. So where do we see them? This is the glycocalyx, this is the yellow one is the cell wall, and the green one is the cell membrane. So these are the three layers. So we will talk about it in the next slide. Next is cytoplasm, which is the fluid filled space. This blue colored structure is the cytoplasm. It contains all the enzymes, cell components and organic molecules. So everything is present in this cytoplasm. Next is the genetic material. It contains the genetic material is again scattered in the cytoplasm, but there is one area where most of the genetic material is present. So that is little dense. So this is your genetic material. So genetic material consists of DNA, which can be single DNA or circular DNA. Now some genetic material is also present like this, small chunks of genetic material. So these circular DNA structures are known as plasmids. So these are called plasmids. So here you see there is no proper organization of the genetic material, but if you think of the eukaryotic cell, you had a proper membrane called nuclear membrane. Inside that you had the nucleus. Inside that you also had a specific region called nucleolus, where you had all the the chromatin thread. So everything was well organized. Here it is not like that. Some genetic material is here, some genetic material is like present in separate chunks, and these are called plasmids. Sometimes this dense area is also given a name of nucleoid. This is called nucleoid. This is not a cell organelle. Please remember, it is just that dense area is given this name. So these genes help in the process of reproduction. The process still remains the same. The genes will do the same thing. The cytoplasm will perform the same function. So I am not going into the detail of each of them now. Ribosomes are present, but they are 70S ribosomes. So where are the ribosomes? These small structures are the ribosomes, and they help in protein synthesis. So the function of ribosomes again will remain the same, just that their size are little smaller than what they were in eukaryotes. Some inclusion bodies are also present, that is, some structures which are not membrane bound, but they store some reserve materials. So here you can see some structures like this. So they help in storing some materials. So for storage of food also you need some structures at least. So they are nothing but these inclusion bodies, like the fat bodies or something. There are also some thin hair-like structures on the cell surface which are called pili, and this pili help to attach to host bodies. For example, bacteria are one of the most common examples of prokaryotes. So bacteria often live inside the body of another organism. Like you see so many bacteria living inside the human intestine. So many bacteria live inside the body of a plant. So they need some structures to attach themselves to the host bodies. So pili are those structures. So you see here, these structures which are coming out from the cell membrane, they are the pili. They will help them to attach to another host body. So these are the structures inside a prokaryotic cell. So now I don't really need to explain cytoplasm, ribosomes or the genetic material in detail, because you already know about all these. So we will only talk about the things which were not present in eukaryotic cell, but they are present now in prokaryotes. So we will first talk about the cell envelope, because this is quite different here. So the cell envelope, as I said, has 3 layers. The first layer is the glycocalyx, that is the outermost layer. This again varies. It is generally composed of carbohydrates. That is why the name is glycocalyx. So glyco is always related to glucose, carbohydrates. It is composed of carbohydrates. They can be a loose outer layer, or they can be a thick, tough and rigid layer as well. So in some organisms, they act as a loose, not delicate outermost layer. That there it is known as slime layer because of its slimy nature. Whereas in some other cells, they are very thick, tough and rigid, and there they are known as capsule. So these are the 2 names given to the 2 types of glycocalyx. The delicate one is slime layer and the tough one is capsule. They ensure protection against other organisms. Since bacteria are very small, minute organisms, they might be eaten up by some other organisms. So glycocalyx ensures protection against other organisms. Their composition varies in different prokaryotes. I mean, the composition of glycocalyx, even though is like mostly carbohydrates, but it varies from one cell to another. The next layer is the cell wall. Cell wall, again, we all know what is the cell wall made up of, what are the layers of cell wall. So I'll not get into those details. It provides structural support. It maintains the shape of the cell as it was in case of eukaryotes. And the third one is cell membrane, which is the semi-permeable membrane, or what we call as selectively permeable membrane. The structure and everything is similar to the eukaryotes. The only thing which is unique to prokaryotes is the presence of mesosomes. So here from the cell membranes, some different extensions or a different layer is formed, which is called mesosome. So it is formed by extension of the cell membrane. So this layer was not formed in case of eukaryotes. So this is something different which happens in case of a prokaryotic cell. So let us quickly see what is the function of mesosome. Since it is an additional layer which is formed there, what does it do? It helps in the formation of cell wall. It also helps in DNA replication. It also helps in processes like respiration and secretion, because here you do not have separate organelles for each of them. Like the process of cellular respiration, in case of eukaryotes, it was handled by the mitochondria. The secretion processes was handled by the Golgi bodies, but here you do not have separate organelles. So all the different tasks need to be handled by these few cell components. So mesosome takes up some of the activities. So this was all about the cell envelope. Now we will talk about the motility of prokaryotes. So can prokaryotes move, or they are static? Of course they can move. However, there are few prokaryotes which cannot move. So they can be motile or non-motile. So let me give you some examples of motile prokaryotes and non-motile prokaryotes. When you talk of motile prokaryotes, most of the spiral bacteria or the bacillus bacteria, they are all motile. So examples of motile bacteria would be the spiral bacteria or the bacillus ones, the cylindrical ones. When you talk about non-motile bacteria which cannot move, most of the coccus or cocci, which are the spherical bacteria, they cannot move. So it depends. Some of them can move, some of them cannot move. So how do they move? So for that they have this structure called flagella. So flagella's wave-like motion helps in the movement of the prokaryotes. Now we have already talked about flagella in case of eukaryotes, but still we want to talk about flagella in prokaryotes, because the structure of flagellum in case of prokaryotes is little different than what we studied in case of eukaryotes. So that is why we are talking about it once again. So here the structure is little different. So here the flagellum is composed of the protein flagellin, and it has three main parts. Filament, which is the longest part. So this entire part is the filament, and it extends from the cell surface to the outside. The next part is the hook, and the third part is the basal body. So these are the three parts. So basal body is the area where it attaches to the organism's cell surface. So that is the basal body. After that, the next area is hook, and then it is followed by the filament. Now when the movement happens, it basically has a motor-like structure inside which rotates. So that motor-like structure actually drives the wave motion of the filament. So that is how the flagella moves. We will not get into the mechanism of movement of flagellum right now, because that is quite complex at this level. But for you should understand that the prokaryotes flagellum are not exactly identical to the eukaryotic flagellum. Thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.